Bismillahirrahmanirrahim, and welcome back to this next video. And this is the ninth video of the chapter 2 of Immunology. From the last couple of videos, we are talking about the neutrophils, about its structure and function, and we are focusing on the primary granules in the neutrophils, because these neutrophils, they belong to the class of the white blood cells that are known as the granulocytes. That means that they contain different kind of the granules. So in the primary granules, I've told you that there are a lot of antimicrobial proteins, a lot of antimicrobial components, and we discussed about the function of the lysozyme, the function of the defensins, the function of the elastase in my previous videos, and I'll share the link in the description. In this particular video, I'm going to focus on another important antimicrobial component, another antimicrobial protein, which is known as the cathepsin. And these cathepsins, they are present in the primary granules, which are also known as the Azurophilic granules. So these cathepsins, they are actually a group of the proteolytic enzymes. They are the endopeptidases. That means they cut within the proteins. And these cathepsins, they are responsible for the protein degradation. They are responsible for the antigen presentation, and they are also very important in the tissue remodeling. Now, when you talk about these cathepsins, they belong to a particular class of the proteases, which are known as the cysteine, aspartic, and serine protease family. Now, these cathepsins, they are actually produced in the inactive precursor forms, which are known as the zymogens. They are also sometimes known as the proenzymes. And when their activity that is needed, so you are going for the removal of a propeptide segment. And when those particular propeptide segments, they are removed, they are going to expose the active site of this cathepsin enzyme, and then they can perform their function. If you look at the targets of these cathepsins, one of the important targets of these cathepsins, they are the cell wall proteins. Now, there are a lot of important cell wall proteins which are responsible for the integrity of the cell wall, and this particular cathepsin, when it goes and it degrades those particular proteins, that is going to compromise the structural integrity of the bacterium. So this is one of the targets of the cathepsin. Another important target, they are the membrane proteins. So the cathepsin, they are also responsible for the degradation of the proteins that are embedded in the bacterial cell membranes. And when they are degraded, that is badly affecting the permeability and functions of the bacterial membranes. And you all know, when the membranes, they are disrupted, the semi-permeable nature of these membranes, that is gone. So those particular things that enter into the cell, which normally are not allowed to enter into a cell. So therefore, they are negatively affecting the bacterial cell when the membrane structural integrity is compromised. Another important component is that these cathepsins, they are going to target the secreted proteins. And what the pathogens with the bacteria, they usually do is that they secrete proteins as part of their virulence factors or as part of their nutrient acquisition. So what these cathepsins do is that they are going to target these bacterial proteins, interfering with the bacterium's ability to establish infection. Let me give you a few examples from this particular class. When you talk about the Staphylococcus aureus, that is a very important pathogen, gram-positive bacterial pathogen. And the Staphylococcus aureus, that produce a particular protein which is known as the staphophane. And this particular staphophane is an important virus protein, which is cysteine protease involved in the immune invasion. So when the cathepsin that degrade this particular staphophane protein, the Staphylococcus aureus cannot evade the immune system of the host, and the immune system can neutralize the Staphylococcus aureus. Another important one from the gram-negative bacteria, you have got the Pseudomonas aeruginosa, a very important gram-negative bacterial pathogen that release an important exotoxin, which is known as the exotoxin A, and this exotoxin A is an important virulence factor of the Pseudomonas aeruginosa. So when the cathepsin that degrade this Pseudomonas aeruginosa exotoxin A, you are degrading the virulence factor of the Pseudomonas aeruginosa, so you are actually controlling the virulence of this particular bacteria. When you talk about the fungi, the Candida albicans is a very important pathogen, fungal pathogen. So what these cathepsin do, that it can also degrade the Candida albicans proteins. For example, the Candida albicans produce a particular adhesin protein, which is very important for the adhesion of these fungal cells to the host cells. So when the cathepsin that degrade this particular adhesin ALS3, the Candida albicans cannot establish, it cannot adhere to the host cell, thereby it cannot establish itself. So this is the function of the cathepsin. So if you like the video, please subscribe to my channel, hit the notification bell, do comment on the video if you want to ask any question, and do share it with your friends. We will continue the discussion in the next video.